Chimera, the first thing you want to do is click browse, and then you're going to find wherever it is in your files that your PDB file is that you want to open up in Chimera. So past students, okay. Actually, I don't want this one. Because I don't want to change what we did. I want to use the copy. There we go. Okay. So, when you get your structure from AlphaFold, this will be like one color in tan. So it'll be two proteins that are in a complex, but it's going to be one color. So it's up to you to color the proteins by whatever. So I'll show you how to do that. So let's first make this all one color. So first I want to hide any atoms. Okay, so I hit all that. Oh, also with Chimera, do you have like a mouse?
Yeah.
Okay. Only use a mouse, and I'll explain to you how to do these different things. So to do this motion, I'm scrolling up and down on my mouse.
Okay.
On the middle thing. To tilt it or to spin it like this, I'm left clicking and I'm dragging. So this allows you to turn it any way you want on its center of axis.
Okay.
Now to do this, zoom in, zoom out again, I'm holding my right click button.
Okay.
And then to move, like say you're super zoomed in, right? And you want to just like move it to the left a little bit, right? If you click the scroll thing in the middle, this allows you to move it on the XY axis, in the XY plane. And basically, if you don't have a mouse, I have no idea how to do all that. I open Chimera on my laptop without a mouse, and like somehow I always figure it out how to maneuver it around, but I don't know the commands like off the top of my head. And then to do what I did, which was select everything, was you have to hold Control and then left click drag, and that selects everything. Or it selects everything that you tell it to select, right? Okay, so let's make this a really boring color, like what it would come in. Okay, so it's all one color now, right? Clear selection. All one color. So when you're working in Chimera, you want to have this thing always open, your model panel. Because basically what you'll do is you'll have like a master Chimera session, and then each one of these will be one of your protein complexes. And it's so you can be in one session and you can flip between different protein complexes, whichever you're working on. And basically like, okay, so I don't want to work with this protein complex anymore, so I uncheck active and uncheck shown, and then let's work on this complex instead. Right? And then this pops up. So which one was it? This one? No. Yes, this one. Okay. So now, how to color. So basically what you have to do is you have to go to Tools, Sequence, and then I want it to show me, this is so complicated once there's so many in here, and they're all named similar things. So this one is called CDC 25A Check 2. So CDC 25A Check 2. Okay, and then because there's 2 chains, so basically a chain means a monomer. So because you'll be complexing 2 different monomers together, it creates 2 chains. So there's 2 CDC 25A check 2s. So I'm gonna control click both of them and say show, and it's gonna pop up both sequences for me. And these sequences are, one is CDC 25A, the other one is check 2. How do I know which one is CDC 25A and which one is check 2? Chimera is not going to tell me. I have to go back to my sequences and I have to just look and see, like, okay, does it end in AVL? Or what is this one? LKKL. I think this one let's see. I think CC25A is the smaller one. 501, 501. They're like the same size. Anyways, I think CDC 25A is this LKKL one, from what I remember from when my student and I did this thing. So anyways, basically what you want to do is you want to highlight the entire sequence, and so that will select the entire sequence on the protein. And then you go to Actions, Color, and do whatever color you want. And I suggest you do, because you want to publish, right? So look up colorblind friendly combinations. That's one thing we have to be mindful of in publications, is choosing colors that are colorblind friendly. So that's like green and magenta, or purple and orange, or yellow and blue. Those are all colorblind friendly combinations. So basically, like, colorblind people may not be able to see those different colors, but when you use those 2 colors in comparison, it's enough difference that they can tell the difference between the 2 colors. Okay, so you select it, choose your color, boom. And then to color your other chain, you do the same thing where you highlight the whole thing, and then color and magenta.
Okay.
And so that's how you do the coloring. And then once you do that, what you'll be doing is you're gonna have to analyze this. So, like, you see here where this strand of pink meets this helix of green. This is what we call a complex interface. And so what you'll do is you will select these residues in this area, and then you'll do something. So you go to Actions, Atoms, Bonds, and you say Show. And it looks like a nightmare, doesn't it? But you also want to say Actions, Color by Heteroatom. And so the next thing you'll do is you'll look for places of contact. So you can do that by looking at it manually, or you can use this thing called Tools. You go to Tools, you go to Structure Analysis, and then you say Find H-bonds. And so you can say, so restrict to models, you only want it to be to CC25A check 2. Only find H-bonds with at least one end selected, so that way you're just looking at H-bonds within the area that you're looking at. If endpoint atom is hidden, show endpoint residue. Yeah, this is good. And then, okay, so this is like hella H-bonds, right? All these black lines. So how do you know what are important ones? Well, anything that's like in between the backbone residues, not important. There's lots of hydrogen bonding that happens in the backbone, because that's how like the structure of the protein is formed. But what we're interested in are the interactions between the R groups, which the R groups are all these things that are pointing out. They're the individual, basically they're like the group that does the action of the amino acid. So, for example, right here, you can see, you can see there's H bonding between this serine, this threonine, and this glutamate. And so this is important because basically this is a phosphatase. And so like phosphatases have serines and threonines that get dephosphorylated, and so that's pertinent. But for you, what you'll be looking for is residues that interact to form, or to residues that interact with each other that are going to interact with a divalent cation. So, or a monovalent cation. So let me show you this. So it says TAS1R2 and TAS1R3 are glucose transporters, and are ATP-gated potassium channels, right? So potassium's a monovalent cation. And so what I would do is I would Google what amino acids interact strongly with potassium ions. So it says aromatic residues like tyrosine and phenylalanine are residues that would interact strongly with potassium. So when you are looking at your structure, you'll be looking for aromatic residues, which are circular ones with rings. So one thing I suggest you do when you are doing your analysis, what did it just do? Is print this picture.
Okay.
This is your 20 amino acids. And when you, so it says that for potassium, the most important ones are phenylalanine and tyrosine. So you're looking for ones that have R groups like this. And so this OHONH2, see how it's there in every amino acid? That's the backbone. So you don't care about that. You just care about what stems off from the backbone.
Okay.
So yeah, you'll just have to have your amino acid structures facing you so that way you know what each one looks like, because to you this probably looks like a bunch of sticks and what the heck, right?
Yeah.
But to me, I can tell this is an aspartic acid, this is an asparagine, this is a serine, this is a glutamic acid, this is an arginine. Like, I can tell those things because I know all my amino acids in my head.
So
So,
what do I do after I identify them? After I identify them, what do I do?
Add them to your spreadsheet. So this is like another column for your spreadsheet, I guess. You'll have to be like, maybe name the column like putative crucial residues or something like that. And yeah, so it's basically going to be like at the interface in which they interact, there's going to be some sort of like cation channel. And so, like, I want to see if I can. So this is orco, right? But this is just a monomer. So how these proteins come together is this helix right here comes together like 4 times. And then because it's got this like flexible area up here, that's like the channel that the ions flow through. So when your complex comes together, you'll be able to see something more clear of like, okay, this kind of looks like something that, you know, ions would flow through. Because your biggest clue is you want the helices to be up and down. Oops. You want them to be up and down because they're transmembrane. So I actually think I have it upside down. There we go.
Should I like download these images? Okay. Should I download these images as well?
Yeah, so you can take like as many screenshots as you want and just like label the screenshots like appropriately. So that way you know what it is and I know what it is when I look at it. And then, yeah, so when you start looking at it, I'm gonna say this. So when you get your PDB file for your first file that you want to analyze in Chimera, send that PDB file to me and I'll look at it in Chimera and like make sure that like I can identify something. And then, because I'm not gonna have you stare at something till you're blue in the face if like I can't even look at it and find something, you know what I mean?
Should I send you all of them that I find, or just like the first one?
Just do the first one and then do any other one that you're having problems with.
Okay. Yeah.
But does all this